Hello again, I'm Vicky. I'm a first-year postdoc at the Schmidt Center, and today I have the pleasure of introducing our next speaker, Maria Brbic. Maria is an assistant professor in computer science and life science at the Swiss Federal Institute of Technology, EPFL, and the head of the machine learning for Biomedicine lab. Before taking on these impressive roles, she did her postdoc in Stanford and received her PhD from the University of Zagreb in Croatia. She has received numerous scholarships and awards already, and most recently, the prestigious SNSF Starting Grant. Maria's research is a wonderful example of what we do here at the Symposium. Her lab works on cutting-edge machine learning methods informed by biological problems and is contributing impressive advancements in multimodal generative modeling for cellular biology. I'm very excited, Maria, to hear your talk, and the floor is all yours.
Thank you so much, Victoria, for a nice introduction. Thank you also, Caroline, for inviting me, and super excited to be here and be part of these exciting 2 days in this workshop. So today I'll tell you about how can generative AI help us to unlock the complexity of cells. So we're in the midst of a genomics revolution that is transforming our understanding of biology at a cellular level. With the advances of single-cell RNA sequencing technologies, we can nowadays measure gene expressions in hundreds or even millions of cells in a single experiment, leading to the creation of these large-scale cell atlas data sets that offer the opportunity to characterize our cells at unprecedented resolution. And not only that we can measure cell transcriptome, we can also measure proteins, epigenome, phenome, and with multi-omics technologies, we can even do multiple measurements in the same time at the same cells. And very latest spatial technologies enable us to study cells in their native tissue context, while at the same time capturing the gene expressions of the cells. At the same time, we are also in the midst of AI revolution. So with the rise of generative AI paradigm, we've seen completely new capabilities of AI system that kind of offer completely new capabilities that were not possible before. And this brings us all to the era of foundation models, where kind of most famous example would be large language models such as ChatGPT that are pre-trained on internet-scale data to predict next token in a sentence, and then can be applied to a variety of downstream tasks. But the question is, how can AI help us to advance our understanding of biology? How can really it brings us closer to understanding our cells, how they function, and what goes wrong on a cellular level in disease state. And our goal ideally is not just to make predictions, but really to use AI to help us to discover new biological mechanisms. And one of the kind of big challenges when you think about biological data compared to, for example, computer vision or natural language processing is that biological data is inherently heterogeneous. So we collect these data sets from different tissues, different donors, different time points, different species, and also we measure different modalities. And the ultimate goal is really to develop an integrative, holistic understanding of biology that captures the complexity of life across different scales. And artificial intelligence technologies really kind of all hold this promise to help us to unify these kind of fragmented views and to discover generalizable patterns across these different biological contexts. And in our prior work, we tackled individually some of these kind of challenges. For example, how can we transfer learn across different tissues, different donors? How can we integrate data sets across different species? How can we analyze data sets across different time points, to kind of mention a few examples. But in this talk, what I want to focus on is more kind of this aspect of different modalities. In particular, combining kind of information from the transcriptome of the cells and their spatial organization. So how can AI help us to learn the relationship between the spatial organization of the cells and their gene expressions? And in the second part of the talk, I want to kind of briefly touch upon how can we leverage kind of these big foundation models that we have nowadays to help for unsupervised inference and kind of to help us to discover what classes automatically exist in the data, for example, what kind of cell types we have in our data sets. So while single-cell RNA sequencing allows us to kind of study our cells at unprecedented resolution, the fundamental problem and challenge is that cells are dissociated from tissue and we lose the spatial context of the cells. On the other hand, we have spatial transcriptomics technologies that kind of capture this that capture organization of the cells in their native tissue context, but the number of genes that we can capture is limited. But can we overcome the limitations of these technologies with computational approaches? And kind of can we get the best of both worlds to really obtain a more complete pictures of how tissue function and understand cell-cell interactions? So ideally, we would like to have kind of spatial context of the cells captured as well as like a full transcriptomic information. So we started this project with the question and idea, how well or can we reconstruct complex tissue structures de novo? So if someone gives me dissociated single cells, can I be able to predict the kind of complex tissue organization? Can I predict how cells get organized in the space? And we call this process of going from dissociated single cells to kind of these tissue structures the tissue reassembly. Because kind of we want to reassemble tissues from these kind of dissociated cells and predict how they get, cells get spatially organized. And conceptually, you can imagine this as kind of AlphaFold for cells, where in AlphaFold we go from the protein sequence to the protein structure. So here what we want to do is we want to go from the expressions of dissociated single cells to these kind of tissue structures. But the question is, of course, to what extent and is this tissue reassembly possible? And of course, the answer will probably will also depend on different biological systems. But given kind of that, if you think about vast cellular heterogeneity, complexity of tissue structures, intricate cell-cell interactions, and also elusive biological mechanisms that underlie tissue formation. And if it is indeed possible, how can we learn it? How can we learn these underlying rules of complex spatial organization? So the good news is that we have like a bunch of existing spatial transcriptomics data sets. So we could leverage these existing spatial transcriptomics data sets to kind of use AI to learn proper priors of how, proper spatial priors of tissue organization. So in order to do so, what we need to do is we need to capture the cellular interactions locally as well as globally across the entire slice. So in particular, what we need to learn in order to capture these cell-cell interactions is how each cell should kind of attend to other cells based on its own location and the gene expression. And we can kind of, in machine learning language, the way we could kind of capture this is using kind of well-known principle, which is a self-attention mechanism. So basically, what we can do is we can learn how each cell should attend to other cell based on the relevance of other cells to a cell's own location and its own gene expressions. So in this way, kind of we can enable each cell to learn how to communicate to other cells guided by the attention rates or attention scores. And secondly, we model this kind of tissue reassembly process as a generative process. So in particular, we model it as a conditional point cloud generation, where the conditioning is applied on each individual point using the gene expressions of the cells. So during inference, ideally, and Joey kind of gave a really nice introduction to the diffusion model, so it was really a good introduction before this talk. But ideally then, during the inference, what we want to do is we want to sample noise, and then these noisy cell coordinates, and I want to condition these noisy cell coordinates on the gene expressions of the cells, and then with a number of steps, I want to be able to kind of go to reconstruct these tissue structures. And these two ideas, to kind of first model cell interactions via attention mechanism, and secondly, the model tissue reassembly process as a generative process, bring us to the framework that we called Luna, and this was work done by my amazing PhD student Yeast and master student Chanakya, who is now a PhD student actually here at MIT. So what Luna does is, Luna leverages these existing spatial transcriptomics data sets with ground truth cell locations. And as a diffusion model, it consists of two main steps. So first step is corruption step. we are going to add noise to the ground truth cell location. We are going to sample diffusion time, and then we are going from these ground truth cell locations to this kind of noise cell coordinates. And then during the denoising process, the model will learn to recover the original cell coordinates through the denoising process. So basically, we are going to learn these cell embeddings using the attention mechanism. And then from these output cell embeddings, we just add one fully connected neural network layer to go to these predicted physical locations, predicted cell coordinates. And then during a training, the model needs to learn to denoise the cell coordinates. Another challenge is basically that when you think about these data sets, that the spatial arrangement of the cells in a tissue, actually it may undergo transformations due to experimental artifacts. While, for example, tissue slices can be arbitrarily rotated and reflected, while gene expressions actually remain unchanged. So this means that we want to basically design the objective function that is invariant to these rotations and reflection. So this means that we want to basically be able, we are not care really about the absolute positions of the cells, but what we care about is about the relative spatial relationships of the cells. So we design an objective function that kind of is based on these pairwise distances of the cells rather on their absolute positions. And then during the inference stage, we sample from the standard normal distribution, and then with a number of diffusion steps, eventually ideally converge to the ground truth cell locations. Luna has also linear time and memory complexity during the inference because we use attention with the linear complexity. So this means it's like a highly scalable. It can infer locations of tens of thousands of cells in just a few minutes on a single GPU. And next, I'll show some results of applying Luna. So first, we applied it on the Murphy mouse brain atlas, where actually we have the ground truth cell location. So we trained the Luna on the 2.8 million cells from one mouse, and then we were interested whether we can kind of reassemble the brain of another mouse. So we applied it to 1.2 million cells from another mouse. And here results of, on the left, I show the ground truth locations. On the right are on the right are ground truth locations. On the left are Luna's predictions. And this is how, for one slice of isocortex region. And here is how the diffusion process happens. So with a number of diffusion steps, model eventually converges to the locations that really highly resemble the ground truth locations. And here, cells are colored based on about 338 different subtypes that are annotated in the data set, but we also see high accuracy if you look at the more finer grained annotations, over 1,000 types, and also cell cluster level, which has over 5,000 types. And overall, Luna's predictions agree well across 11 major regions that are identified in the ABC atlas, despite their distinct structural characteristics. If you look within a particular cell subtype, so for example, if you look at, this is just an example of glutamatergic neurons, we further look whether basically Luna is able to infer this kind of spatial distribution of gene expressions correctly. So these are examples of two spatially variable genes, RXFP1 and RORB1 gene, or RORB gene, where here on the top are ground truth locations, on the bottom Luna's predictions, and this RXFP1 gene is actually highly expressed in this lower part of the region, and here a bit mildly expressed here, and this is actually correctly captured by the model, as well as also spatial distribution of the RORB gene. This is also the case for some of the non-neuronal types. So this is an example of ependymal, astrocyte ependymal cells, where there is a small group of cells expressing the CFAP206 gene, which is actually also correctly captured by the model. And given the kind of high similarity of the two brains, we were also next interested to see whether model could generalize if we, for example, exclude certain major cell class during training. So we excluded we went to make the task most challenging. We actually excluded, we went to the most coarse-grained annotation. So on there are 34 different major classes defined in ABC Atlas. And here, basically, during the training, we train on all cells from animal 1, but we completely exclude, so here is just an example of glutamatergic neuron cells in layer 6B. And then we were interested to see whether model can still be able to correctly predict where to locate these cells in another animal, even though this cell class has not been seen during training. Indeed, we found that the model correctly positioned this unseen cell class in the spatial architecture of the tissue. And again, if you look at the spatially variable genes, for example, TSHZ2 gene and SYT6 gene, it was able to correctly capture the spatial distribution of this gene within this cell class. This is also the case for other cell classes, so this is just an example of another cell class, which was the excluded vascular class, which has also very different distribution across different slices and was actually correctly captured by the model, as well as the expression of the GFAP gene, which is a spatially variable gene for the vascular class. So Luna is unique in terms of kind of learning these spatial priors across multiple slices, but one could potentially apply some of the existing methods to this task. For example, the most closest methods would be methods such as tangram. So these are methods that map dissociated cells to a reference data set. But the limitation of these methods is that you have to have exact spatial reference to map your dissociated single cells to your data set. Another class of methods are methods such as NovoSpark, where you do kind of also this kind of de novo reconstruction, but they are based on the assumption that if two cells have similar gene expressions, they should also be spatially close to each other, which is not satisfied in complex tissues. And actually, the core idea in Luna is that we try to learn these spatial priors using AI methods. So we compared Luna on this kind of smaller mouse cortex dataset because some of these methods are also not very scalable. If you don't have any prior information put into these models, Luna is actually the only method that is able to correctly infer the spatial priors of the tissue. Here the performance is measured as Spearman's rank correlation, where we order cells based on their similarity and compare closeness and then compare to the ground truth ordering. If we put some prior information about the reference slice for these baseline methods, which Luna does not use, we still see a large improvement over existing methods. So, and next, we were actually wanted to really apply Luna to infer the locations of the single-cell RNA sequencing atlas. So in particular, we take the single-cell central nervous system, single-cell RNA sequencing atlas. Of course, for this, we don't have ground truth cell locations. So in particular, the good thing about this particular atlas is that we could estimate the ground truth because there is star map atlas, which one can integrate with the single-cell RNA sequencing atlas. We don't use it to train Luna or anything. We just use it to validate whether these predictions would be correct. So this is the estimated ground truth that we obtain by integrating this single-cell RNA sequencing atlas with the star map atlas. So these are estimated locations of the single-cell RNA sequencing data, and these are predictions that we obtain by training Luna on this Murphy's mouse brain atlas and then applying into single-cell RNA sequencing data. Again, we see a good resemblance between the estimated ground truth and Luna's predictions, also on the broader classes, and here again, I visualize how the process happens in Luna. If you look again on the gene expressions, for example, the model is able to correctly capture the expressions of some of the spatially variable genes, for example, IQGAP2 gene, which shows downregulation across the Ammons whole region. It's kind of this downregulation is correctly captured by the model, as well as, for example, MIF-TC gene, whose expression is minimal in the CA2 region, but then it's substantially upregulated in isocortex and olfactory areas. So we can now also use Luna, for example, to infer the expressions of genes that have not been seen during model training, because now we can infer this for the single-cell RNA sequencing atlas and look at the spatial distribution of these unseen genes across different regions. And next, we were also interested whether Luna can have to overcome limitations on some of other existing methods. For example, yesterday we heard about SlideText from Fei Chen's lab and Evan McCosco's lab. And SlideText enables this profiling of single-cell and spatially resolved transcriptome, but the limitation of this technology is that many nuclei lose spatial information during the barcoding process. So we asked the question, can we now use Luna to actually predict the tissue locations of the nuclei that have been lost during a cell profiling with SLITEx? So here we applied it for the human melanoma data set. So these are spatially mapped nuclei with SLITEx. And then we enrich this data set using Luna by predicting the locations of these spatially mapped nuclei. So Luna successfully enriched this data set from 4,800 to 6,500 cells. If we look at, in particular, like 2 tumor cells that are placed into 2 tumor subpopulations that are placed into spatially distinct compartments. So these are spatially mapped nuclei with SLITEx. Here are nuclei mapped by Luna. And this one group of tumor cells is expressing the CLU gene, other is expressing the DISC2FP1 gene, and this is actually also correctly captured by the model. We next investigated whether this Luna-enriched SLITEx dataset can help us to increase the power to detect spatially variable genes in the human metastatic melanoma tissue. So we detected spatially variable genes using only an original sample, where we were able to find about 400 genes that are detectable both in the original dataset, just using original dataset, and also Luna-enriched dataset. But using Luna-enriched dataset, we were also able to detect additionally about 400 additional spatial variable genes. If you look at the gene pathways that are enriched in this set of genes, these are indeed, for example, the most significant pathway is the pathway regulated by the MYC oncogene. We also applied it to other SLITEx data. So this is, for example, for the mouse embryonic data set, where it helped to enrich the data set by twice. So actually, Luna can also be used potentially, to think about kind of knocking out the effect of knocking out certain gene and kind of going potentially towards this kind of virtual tissue models when we can kind of knock out certain gene and then ask what happens to the spatial architecture of the tissue. So what we did is we just trained the Luna on the whole data set on the Murphy mouse brain atlas from an animal one. And then during the inference stage, we were interested to see what happens with the predictions of the Luna if I knock out one gene per study, and then measure the effect basically of knocking out this gene on the prediction and the spatial architecture of the tissue. So in particular, what we can do is we can, for every study, if I knock out certain gene, I can then measure the effect knocking out this gene has on the spatial structure. So basically, I get the maximum performance if I include all genes, and then I measure how much excluding this gene is affecting my performance. And then I can identify these genes that have the highest effect on the spatial, on perturbing actually the spatial structure of a tissue. And we can do this, for example, for different also cell subclasses. So here I show example for 5 different subclasses, and here are genes that have highest effect on the spatial architecture. So for example, these are indeed genes that are very important, crucial genes for the cell identity and function of these cell classes. For example, Claudin 11 gene is oligodendrocyte-specific protein, and previous work has shown that mutant mouse that lack this protein exhibit central auditory deficits and the neurotransmitter imbalances. We can also do this for pairs, like basically seeing what happens if we exclude pairs of genes. But of course, the number of combinations in that case is very large. But what we do is we use existing receptor ligand databases and only focused on a certain subset of possible pairs. And in the last part of my talk, I want to also quickly talk about how can we, now in this kind of era of big foundation models, how can we actually do something like unsupervised transfer? How can we use these big foundation models that people have been developing to actually tell us, what kind of categories exist in the data in a fully unsupervised fashion? So if you think about current paradigms in which we use these existing foundation models, they still majorly require supervision. So the standard way, if someone gives you this foundation model, we kind of fine-tune it on the task of interest using labeled data. So this is just a toy example where someone gives me large pre-trained foundation model, and then I have my labels, and I want to solve a new task. I would kind of add a linear classifier to distinguish between different cell classes. But as we've seen yesterday in Jennifer's talk as well, is basically that in many cases in biology, we actually don't have these labeled examples. So we cannot kind of just add a linear classifier to solve our task of interest because we lack the labeled examples. We cannot have abundantly labeled examples for every possible class. Ideally, we want to use these kind of big models to tell us what exists in the data in a fully unsupervised manner. So alternatively, in machine learning community, people do like zero-shot transfer on the task of interest using instruction set, where you basically, the most famous example is OpenAI's CLIP model, where you have this image encoder, you have the text encoder that encodes this kind of class descriptions that you expect to see in the data, then you match them. But in this case, you still kind of require to know which kind of classes you expect to see in your data. And this is still not kind of suitable often for biology and for biomedical applications. So the question we ask is, how can we infer these underlying classes without any supervision? So if one gives me, large pre-trained foundation model, and this is like, we think about this very generally, it could be kind of computer vision foundation model trained on real-world images. It can be single-cell foundation model trained on single-cell data. It could be, histopathology foundation model trained on histopathology images. How can I, in an unsupervised way, say, what classes exist in this data set? Of course, the trivial solution would be I just run the clustering to discover these set of classes, but the question is, can we do better than that? And we call this unsupervised transfer because we want to leverage these foundation models to, in an unsupervised way, transfer knowledge to a new task that I'm interested in to solve. And the key insight to solve this problem for us was that if you think about how we currently use these models, we use these models. The key insight is basically that linear models in representation space generalize well. So that's why we can just add a linear classifier on top of this pre-trained representation, and typically we get very high performance if we have the labels. And what we observe indeed that if we look kind of at the agreement of with the ground truth human labeling of the possible labeling, if you imagine kind of I give you labeling, you can generate many possible labelings of these data sets. But if I measure the agreement with the ground truth human labeling, the generalization of error of the linear classifier will be lower if this is kind of labeling agrees better with the ground truth human labeling. And this kind of idea motivates us to develop a framework that we call Turtle, and this is work done by my PhD student Artyom and Yulun. And the key idea is that basically we want to rephrase this unsupervised learning problem from the representations of foundation models as a problem of searching for such labeling so that linear classifiers will generalize well in this representation space. So I can take my data set of interest. Again, it can be whatever foundation model or whatever data set it is. So this is just, for example, these kind of real-world images. I can embed it to one or multiple foundation models. If I have multiple representations, I can leverage multiple foundation models. And now I can basically solve this problem by finding such labeling that will give me low generalization error of a linear classifier. So we formulate bi-level optimization procedure when in the upper loop we basically search, we generate the possible labelings, and then in the inner part of the optimization we evaluate how well does that labeling, how well can linear classifier generalize on that labeling, and then we optimize over possible labelings of a given data set. So I won't go into many details. I'm happy to chat later, but basically searching for these labelings requires solving a discrete optimization problem over all possible labelings. So instead, we resort to continuous optimization via relaxation. Instead of doing discrete search over labelings, we do continuous search over parameters of the task encoder. And we can also show theoretically that the labelings that will be found by this optimization procedure under some assumptions are the labelings that maximize margin of a linear model in the corresponding representation space. So Interestingly, this kind of idea, although very simple, it actually allows us to leverage now any foundation models, train linear classifiers on top, and search for the possible labelings. We show that it achieves state-of-the-art unsupervised performance across various benchmarks, so it's available on the papers with code. You can use it with any, your favorite foundation models. So this is an example of the ImageNet dataset, where actually the state-of-the-art method, as well as also many other datasets, including, for example, pathological dataset of histopathology images like patch chameleon. We went also a step further to compare it to the zero-shot transfer, like, for example, the CLIP model that actually uses information about the classes, while Turtle is fully unsupervised. And we show if we run the Turtle in the same embedding space as the CLIP, we have the same performance. But if you just add another foundation model, which is much cheaper than getting the labelings, for example, from the CLIP model, you just add another representation space. For example, here we just add a Dino model, then model significantly outperforms CLIP model across this 26 benchmark data set, achieves a large improvements. We can also apply it to single cell data. So this is an example of using existing foundation models for a single cell, where you can do this unsupervised transfer to discover, for example, different cell classes that exist in the data. But I would say disclaimer here is that the core idea of the method is that you assume that the representation space of foundation model is strong enough, which is sometimes case for these foundation models that we have for single cell, but sometimes still I think we have a large space to improve these models. So this is just one slide. So in the very latest work, we also show that we can do unsupervised adaptation of these big foundation models. So we can even fine-tune them in an unsupervised way. So instead of doing this kind of linear classifier in the inner loop, we kind of perform in-context learning in the inner loop. And now we show that basically this kind of framework that we call joint inference, that was just presented as iClear, it can make predictions simultaneously for all inputs in a given task. And we show that this is applicable to, for example, large language models, vision language models, and achieves state-of-the-art performance on the difficult math reasoning benchmark compared to even this unsupervised ICL can even outperform supervised ICL on these math reasoning benchmarks. And finally, I would like to thank my amazing PhD students whose work I presented here. So in particular, Yeast, who worked on Luna, Yulun and Artyom, who worked on Turtle and this also unsupervised inference framework. And happy to take questions.
Thank you so much for an amazing talk, Maria. Questions?
Thank you. Fantastic work. Just a question about Luna. If I understand well, it's a generative process that is stochastic. So if you run it 10 times, you get 10 different images. Could you comment on the diversity that you get, or is it always consistent?
No, it's not always consistent because it's a generative model, right? So of course, if you run it different times, you will see some variation. Cells would not be exactly positioned at exactly same locations, but still this kind of spatial distribution that I was showing would be preserved. So in terms of maybe, two cells will be kind of switched locations but these kind of results that I was showing, they are kind of consistent across different runs.
Okay. Thank you.
Hi. Hi there. Thanks a lot for a great talk. I have a question on Luna. So what are the limitations of Luna, and what are some of the future directions that you see?
Yeah, so first, the major limitation is actually that tissues are not 2D, right? So for single-cell RNA, like ideally one would actually be able to embed any single-cell RNA sequencing dataset in the 3D space, right? So here, for example, for single-cell RNA sequencing data, we do this kind of integration, with the star map atlas. We have this, we know from this which 2D slice the single-cell RNA sequencing data comes. But ideally, one would apply this to 3D data. But I would say this is not really a limitation on Luna because the core idea would be applicable to the 3D case, but we just don't have the good data to train this model on the 3D data. So the objective function and everything, we can immediately apply it for 3D case as well. But another limitation is that, and what we're looking now further is basically that currently, so when we train the model, we you assume that you measure, for example, existing spatial transcriptomics data sets, that you capture this, that basically, for example, for the merfish, it's okay because we have the same gene panels, but ideally you would kind of be able to train it across like different spatial technologies. Then we would need to kind of be able to capture also like different gene panels that can be measured across different data sets. So this is another limitation of the method.
So much. We sadly have to stop at the 2 questions. Thanks again.
Thank you.
Please applaud Maria again.